Production starts with growing viruses or fermenting bacteria. Very small quantities of these microorganisms are caused to multiply in order to harvest extremely large quantities. From just a few cubic millimeters of solution, it is possible to produce enough virus or bacteria to develop millions of doses of vaccines. Each microorganism requires specific conditions to reproduce. Microorganisms are grown in special, complex growth media that may contain up to 30 different ingredients. A wide range of parameters must be taken into account: temperature, pH, oxygen rate, sterility, homogeneity, and so on. Each factor is important. The slightest variation in the culture media may compromise the results. Depending on the vaccine, cell culture takes from 2 days to 3 months. Following harvest, the next step is purification, which eliminates any trace of culture media and any other impurities. Inactivation takes place next. This step is both extremely delicate and critical. Inactivation consists of destroying the pathogen's ability to cause disease while making sure they will still be able to induce an immune response. At the end of this phase, one obtains the very essence of any vaccine, its antigenic valence. Stabilizers, and in certain cases a preservative, are added. The quantities are extremely small, yet they're sufficient to guarantee that the vaccine will remain stable, potent, and effective. Sometimes an adjuvant is also added to enhance the immune response. The next step consists of filling syringes or vials with the vaccine. Certain vaccines, such as the measles, rabies, and yellow fever vaccines, are not stable in liquid form. They are put through an additional step to stabilize them. Freeze-drying removes moisture from the frozen vaccine under vacuum conditions and at very low temperatures. The product is now a powder. When it's time for the vaccine to be administered, it will be reconstituted by combining this powder with a diluent. Of visual inspection, the human eye is aided by electronic cameras that detect and identify even the smallest defect. Final tests are performed by the vaccine manufacturer and then again by the health authorities. The vaccines are then packaged and ready for shipping. Once again, certain precautions are absolutely necessary. From the moment the syringes and vials are filled, up to the time the vaccines are administered to patients, maintaining the cold chain is of the utmost importance. Vaccines are fragile biological products, and their potency can be lost if they're exposed to light or to temperatures that are too warm or too cold. To guarantee that vaccines will offer protection against disease, they must be stored at a constant temperature, between 2 degrees Celsius and 8 degrees Celsius. Cold rooms, refrigerators, and isothermal packaging ensure that they will be protected during each phase of transportation and storage. A monitoring device is included in each shipment to ensure the temperature is maintained throughout. Even the slightest variation in temperature is indicated. It is challenging, but it's also very exciting. At the end of the vaccine production cycle, the lives of millions of people are protected against many different diseases, some of them fatal. Today we have vaccines that offer protection against 26 different diseases, such as tetanus, tuberculosis, polio, and rabies. Tomorrow, we will be able to fight other diseases, ones for which a vaccine has not yet been developed, such as dengue fever, hospital-acquired infections, and perhaps one day, malaria and AIDS. Vaccination is one of the most innovative fields of medical research, and it has many more fascinating developments in store for us.